Hello everyone. So in this session we are going to study about the female reproductive system. Yesterday we talked about the female reproductive system, only the ovaries we studied, where will be the ovary, and then the fallopian tube, the function of fallopian tube, and the uterus, the layers of uterus. These are all the things we studied yesterday. So in this session we are going to study the next thing that is related to female reproductive system. So as I told yesterday, the female reproductive tract or this system, it not only synthesizes the egg but also it helps in the fertilization, it gives site for the fertilization, and then this system, it develop the embryo inside it, and after the young one, after the parturition, after the giving birth to young one, it also nourishes the young one. So based on that nourishment, we will learn some structure related to that, how the young ones get nourished by the mother. So that is by means of mammary gland. Mammary glands are present both in male and female. In males it is rudimentary, but it means it is non-functional, but in females it is functional, it becomes functional to give birth to nourish the young ones. So we will see the structure of mammary gland here. So it comprises glandular tissue and fat tissue or fat cells. So this is the fat content which is present in the mammary gland, and each mammary gland consists of the lobes. Lobes are 15 to 20 in number, each lobe having number of clusters. I'll talk about that. First let me draw this, let me complete this structure. Let me label these parts also. So this is the rib, and these are the muscles which is present in between the ribs, and these are the fat covers the mammary gland, and these are all the lobes. 15 to 20 lobes are present in each mammary gland. Let me label that mammary lobe. Each lobe consists of cluster of alveoli. Let me label that. This is mammary alveoli. Two alveoli we have to mention because we have written alveoli here. So the alveoli, the cells of this is the alveoli. The cells of alveoli secretes milk. It secretes the milk to the lumen of alveolus. From there, through the mammary tubule, through the each alveoli having it get connected with the mammary tubule. Through the tubule, the milk conducts, it passes, and then just draw the same structure in these lobes so that it will be a complete one. And then the mammary tubule. So here the each alveoli, when the alveoli secretes the milk, it conducted through the mammary tubule. Okay, so that tubule, each tubule get connected with one another, and it opens to mammary duct. So this is the mammary duct. Here will be the mammary duct. Here will be the mammary duct. What it is doing? It is collecting the milk which is coming from the tubule, mammary tubule. Okay, mammary tubule, it receives the milk from the alveoli. And then each tubule, duct, it opens to a wider ampulla. It means from here to here the milk should passes. So that pathway we are showing here. So this is the ampulla, wider ampulla. That wider ampulla, it opens to lactiferous duct. This is the lactiferous duct. That duct, through the duct, milk sucked out. The edge point or edge region of the mammary gland is nipple. So this from this nipple or from the lactiferous duct, the milk sucked out. The milk is for nourishment of young ones. So this is the structure of mammary gland. So this structure they have asked again and again for 5 mark. Refer this and follow this. So with this we will move on to the next concept, that is gametogenesis. Gametogenesis is the process of synthesis of or formation of gametes. So here we are in human reproduction. Humans are unisexual, both male and female are differ, so that the gametogenesis also 2 in number, or it is of 2 types. One is spermatogenesis and then oogenesis. So we will start with the spermatogenesis now. The spermatogenesis is the process of synthesis of sperms from testis. Testis is the male reproductive system or male reproductive organ where the sperms synthesizes, and from the testis it conducts through various parts, and through the urinogenital organ it get released. So first we will speak about the spermatogenesis now. The spermatogenesis, as I told you, the synthesis of male gametes or the sperms. So in the before last session we were talking about the structure of testis. In the testis there are a pair of testes in the extra abdominal cavity. The testis is enclosed or embedded in the scrotum. The scrotum is a pouch like structure having very thin or soft muscles. The testis is covered by a thick layer, thick tissue. That tissue name is tunica albuginea. The tunica albuginea it get infoldings. That tissue, when it get infolds inside the testis, it forms the compartments. That compartments are known as testicular lobule. Are 200 to 250 testicular lobules are present in one testis. So each lobule, each compartment comprises 2 to 3 seminiferous tubule. So I need that seminiferous tubule now, because we are in the synthesis of sperms. Seminiferous tubule. So the seminiferous, when this process starts, when a human male body reaches to puberty. When he reaches to puberty, this whole process starts. The seminiferous tubule is made of, or it is lined by, germinal epithelium. So it is lined by the seminiferous tubule lined by germinal epithelium. So, the germinal epithelium is of two types. One is germ cell and one more is Sertoli cell. Male germ cell and Sertoli cell. Male germ cell it helps in the synthesis of sperms. Sertoli cells nourishes the sperms. So, consider this is one germ cell. Only one germ cell I need here only for the explanation. I will take this germ cell now. So, this germ cell first it undergoes multiplication. So, we have started the spermatogenesis now. Concentrate this. The germinal epithelium, one of the germinal epithelial cell, that is germ cell, male germ cell, it is undergoing multiplication or it is taking the mitotic division. Why mitotic division? Mitotic division is the equational division where the chromosome number won't reduces. Only to increase the germ cells number it undergoes mitotic division. So, here we have number of what is this? Now we are going to start giving a name to these cells now. This is the Spermatogonia. Okay. So, this Spermatogonia, what is the function of Spermatogonia? Not function, it is from where the Spermatogonia releases from the germ cells. When the germ cells takes the mitotic division, when it start multiplying in its number, we got the cells known as Spermatogonia. So, as from here I will write the Spermatogonia. One of the Spermatogonia I am taking here to explain you. So, this Spermatogonia, it grows, it increases its content. When it increases its content, it becomes bigger. Okay, that is nothing but, so this Spermatogonia, what it is happening now, it undergoing the or it is taking the growth phase. Sorry, it is going to the growth phase, means the Spermatogonia increases its content so that it become bigger in size. So, that bigger sperm cell, sorry not sperm cell, that bigger cell is named as primary spermatocyte. So, the primary spermatocyte, so how the primary spermatocyte came? When the Spermatogonia, it attains the growth. When it increases its content, it becomes the primary spermatocyte now. Now, this primary spermatocyte, it takes first meiotic division. This all is happening in the seminiferous tubule. Through the seminiferous tubule, the sperms releases. The first meiotic division is nothing but reductional division. So, the reductional division is nothing but decreasing the chromosome number. The chromosome number at the primary stage or in these stages, the chromosome number will be diploid. It means 2n in number. When the primary, here also the chromosome number will be diploid. When this primary spermatocyte attains or when it undergoes when it takes first meiotic division, it reduces its what? Chromosome number. So, what we are having here, when this primary spermatocyte undergoes meiotic division, the two resultant cells will be the resultant product. So, these two cells are known as secondary spermatocyte. So this is the secondary spermatocyte where the chromosome number has reduced. Alright, now this secondary spermatocyte, again it takes the second meiotic division. When it takes the second meiotic division, the resultant cells will be 4. What will be the cell name? So here we are going to call this as spermatids. The chromosome number will be half of the original cell. Okay, what we are having here? We are having the spermatids. Alright, from where it came? It came from the secondary spermatocyte. How it increases its number when the second meiotic division takes place in the secondary spermatocyte? We got 4 spermatids. So we are describing only one germ cell. Just calculate 200 to 300 seminiferous tubules. Each seminiferous tubules having number of germ cells, all germ cells releasing number of spermatids. So these spermatids are non-motile, spherical, circular structures. It has to develop further. The spermatids, it has to redevelop, reconstruct, you can say it. So construct, and it has to transform. The spermatids transform, and it takes and it become spermatozoa. So this is not the sperm, this is not the structure which is ready for the fertilization. It has to transform, it has to change its structure. How it gets changes? So these are all the spermatids. As I told you, the spermatids, it will change into spermatozoa. That mechanism is known as Spermatogenesis. Spermatogenesis is the process of transformation of spermatids to spermatozoa. Spermatids are non-motile spherical structure. Spermatozoa are nothing but sperms. Sperms are highly active, motile, and having tail and little bit longer or lengthy structure. Okay, that I will explain you. If I draw the structure of sperm, you will come to know what is the exact structure of sperm or spermatozoa. So, how it transforms the spermatid, what are all the changes takes place in the spermatid, after the spermatid, how the sperms attain its natural structure, how it become ready, how it will be ready to travel from one region to other region, we will see in the next session. Till that, what you have to do, you have to learn all these concepts. Yesterday and day before yesterday, I didn't give you the homework. Just write the homework today and show it to me. The first question of the assignment is, draw a neat label diagram of female reproductive system. The second question is, draw a neat label diagram of male reproductive system, and these two are the 5-mark question, and also draw a neat label diagram of mammary gland. These 3 questions you have to write, only diagrams I am telling you, draw it and submit it. Thank you.